Welcome to CEC EduSet lecture. I am Dr. Amit Bhattacharya from Department of Zoology, Ramjas College, University of Delhi. So this is the second part of the lecture where I will be discussing on the antimalarial drug resistance, the basics and the emergence. Now before going into the antimalarial drug resistance, we should understand the basic cycle of the plasmodia, what happens in the host system, especially the human host. So we know that in the mosquito the sexual part of the cycle happens, but in the human host the asexual part of the cycle happens, where it is being divided into two major parts. One is in the liver, the other one is in the red blood cells. So once they infect the liver cells or the hepatic cell, the liver schizogony starts taking place, and once the liver schizogony ends, they start infecting the blood, so the erythrocytic schizogony start taking place. And from the blood, finally the gametocytes are being formed, which are being taken by the mosquito bite, and the further sexual cycles start taking place in the mosquitoes. So this is a diagrammatic image which is showing the basics of how exactly the various stages are formed into the two hosts, which is the humans as well as the mosquitoes. Now, the treatment for the malaria has been basically targeted into four major steps. First, it has been targeted to the human liver stages, because in the human liver stages the tissue schizogons are formed over there. So if they are affected, then what happens is a major part of the plasmodium infection is being neutralized over there. The next one comes is the human blood stages. So the drugs infect the targets these blood schizogons. Apart from that, the various other stages. The third important stage which is being targeted is called as the gametocyte stages. So these gametocytes which are formed once the blood stages are over, so these gametocytes are being targeted. And finally the liver dormant stages, because in many of the species of plasmodium, especially in the plasmodium ovale and plasmodium vivax infection, there is a presence of a liver dormant stages. So what happens is these particular stages go and become dormant in the liver cells. After many years, there may be a resuscitations of this particular disease condition. So these are basically because of these dormant stages which are present. So these dormant stages are basically targeted with the primaquine drug which is available. Now, this is the timeline of the drug which are being used for the malaria and their introduction, which is shown over there. One of the initial drug which was discovered in the 1800 was called as the quinine. Apart from the quinine, later on subsequently in the 1940s and 50s, the chloroquine came into the picture, then the various other drugs, and one of the most recent drugs which has come up over here is called as the artemisinin. So artemisinin combination therapy is one of the most important therapy which is available for malarial infection right now. Now these drugs are being divided and scanned down into various forms, which includes the antimicrobial, the tetracyclines over there, then the antifolates which are there, then comes the artemisinin drugs which are there, mefloquine is there. So these have been characterized and divided into various forms over there. Now these are the structure of few of the major important antimalarial drugs which are there. So as I told you, the artemisinin has formed one of the important class of drugs which are being recommended and used across the globe. This is another set of the images which shows the structures of these antimalarial drugs which are available in the market, like the chloroquine, artesunate, mefloquine, then the pyrimethamine, sulfadoxine. So these are the various common antimalarial drugs which are there and recommended by prescribed by the physicians. Now, how fast these drugs kill the parasites? Now it has been found that the tetracycline takes the maximum time in killing the parasite, then comes the sulfadoxine pyrimethamine, and the artemisinin class of drugs are the most potent form of the drugs which kills the parasite in a very short interval of time. So artemisinin has formed one of the major form of the guidelines and the recommendation for these antimalarial arsenals. Now, in the 1967, for the first time, the WHO defined the drug resistance. So, what is defined is, it is the ability of the parasite strain to survive or multiply despite the administration or absorption of a drug given in a dose equal to or higher than the recommended, but within the tolerance of the subject. So, that is how the drug resistance definition came into the picture. Some years later, it was further modified, including the sentence which says that the form of drug active against the parasite must be able to gain access to the parasite or the infected erythrocyte for the duration of the time necessary for its normal action. Now, if we see, most of the data related to the antimalarial drug resistance and the infections are being uploaded in the National Vector-Borne Disease Control Program website. So, you can check the website over there, in which the drug resistance across the India status and the various data are being provided over there. So, they also give you the mapping of the various centers, how exactly the data is coming up from the various regional center across India, and how they are giving a data that how the resistance is being spreading up. Now, this is a diagram which is showing the complex interplay of the various factors which cause to the emergence and the spread of the resistance. So, we know that in the cause and the spread of the resistance, the antimalarial drugs plays a very important role, the migration of the population plays an important role. Then there are various factors like the self-administration or the subtherapeutic doses which takes place into the consideration. Apart from that, there are ecological consideration which also comes. They also play an important role in the spread of the resistance across the regions. Now, some of the important factors or the key factors which have been noted down by the WHO, which results in the emergence of the drug resistance parasites, includes the inadequate treatment, which is the subtherapeutic doses or the suboptimum doses, inappropriate self-treatment and the mass administration, long drug half-life which is there, and the use of the conventional antimalarial monotherapy. So, if we see nowadays, most of the drugs against the parasites or bacteria, fungal infections, they are not monotherapy, they are coming up in a combination therapy way, because it has been found that the monotherapy is more susceptible for emergence of drug resistance to these particular things. Now, this is a diagrammatic representation which is showing how exactly the spread is happening from a primary infection into a secondary infection. Apart from that, there is an inadequate presence of the drugs pressure over there, or it may be the drug pressure is of higher quantity. Now, this is again another diagram which is showing the decline response to the antimalarial drugs. So, we can see the mefloquine, quinine, the sulfadoxine, pyrimethamine and the chloroquine. So, we can see over the years how these particular drugs have come into the load of the resistance. So, the chloroquine came, then the sulfadoxine, pyrimethamine, quinine and the mefloquine. So, most of the antimalarial drugs, the resistance have been developed by these particular parasites. Now, this is an image which is showing the transmission of these resistance, how exactly these resistance happens. So, these majorly happens because of a delayed response over there, and subsequently due to this delayed response there is a delayed diagnosis and the treatment which happens. So, the drug pressure remains for a larger period of time, and thus the parasite gets a time to develop a resistance against that particular drugs. Now, let us come into the first drug resistance which came into the picture was for the chloroquine. So, chloroquine, we know that it was a synthetic drug which was developed in the 1930s, which revolutionized the malarial treatment regions. Now, the chloroquine was one of the most widespread drugs which was used in the early 1950s until 1990s over there. Now, because of this widespread use for more than 10 years, there is a development of mutation within the Plasmodium falciparum which has been reported. So, there are various point mutation which has been reported in the Plasmodium falciparum, which has led to the chloroquine drug resistant strain development over there. Now, the major resistance started developing independently in the Cambodia, Colombia and the Thailand region over there. The next drug resistance which got developed was the combination drug which was there in the market, which is called as the sulfadoxine pyrimethamine. We know that the sulfadoxine pyrimethamine is a combination drug which affects two of the vital enzymes within the parasite. One is the DHFR and the other one is the DHPS. So, it majorly affects the folate metabolism in the parasite, thereby leading to the parasitic death over there. Now, a combination of these drugs which replaced the chloroquine also came into the drug resistance category later on. So, what happens is there was a high frequency use of these particular drugs, especially in the malarious regions or a region where there was a high transmission rate. Now, currently the drug which is being used is called as the artemisinin based combination therapy. However, very few reported cases have been reported against these particular drugs. Now, why exactly the artemisinin is so not available at each part of the countries? Because of its high production cost actually. So, the artemisinin, because of its high production cost, it's not widespreadly available in the major endemic regions of the globe. So, thereby it is becoming difficult to fight against the spread of this particular disease over there. Now, this is a tabular representation which is showing how when the drugs came into the market and how the distribution went into the picture and in which year the drug resistance happened for that particular drug. So, we can see the drug resistance took the maximum time in the case of the quinine. So, we know that the quinine is a naturally occurring product from the trees. So, about more than 270 years it took for the parasite to develop a resistance against the quinine, while most of the other drugs are the synthetic drugs. Now, the mechanism of resistance is majorly lying in the gene mutations which are present. For example, in the case of the chloroquine, there is a gene mutation which is called as the K76T gene mutation which happens. So, this mutation happens basically of a point mutation of a change of a amino acid over there. Then subsequently there are various mutations, for example, the mefloquine which occurs because of another different type of mutations over there. Now, this is a diagram which is showing that how exactly the spread of the antimalarial drug resistance happens. So, we can see over here, especially it happens in the South Asian regions. First the chloroquine came into the drug resistant picture, then the sulfadoxine pyrimethamine, then the mefloquine, and recently some of the artemisinin reports have come into the picture, but still the scientists across the globe are checking the results to confirm the drug resistance against the artemisinin. Now, what are the key drivers of these antimalarial drug resistance? Now, this is occurring because of the unusual genetic structure of the malarial parasites in the regions which are known for the antimalarial drug resistance. The second major key point is the use of the substandard drugs or the fake drugs or the counterfeit drugs across the market. The third important point is the unregulated or the poorly administered antimalarial drug usage, and the final one is the artemisinin drug uses without using a combination therapy in those particular patients. So, these particular points are leading to the development of the antimalarial drug resistance in the patients. Now, in the year 1955, the WHO launched a very ambitious program which was called as the Global Malaria Eradication Program, in which the DDT was spread across the various regions which were reporting high number of malarial cases over there. Now, it has been found that the South Asian region and the South American regions are the one in which the chloroquine resistance start taking place over there, and subsequently chloroquine resistance in P. falciparum was observed, and it was first observed in Thailand in the year 1957, and during across the same time in the Colombia-Venezuela border also in the year 1959, the chloroquine resistance was reported. So this is a diagram map which is showing that how exactly the spread of the chloroquine happened. The chloroquine resistant forms of the Plasmodium falciparum, which started occurring in the Thailand in the year 1957, how it spread across the South and the Southeast Asia regions, and subsequently to the sub-Saharan African regions and the South American regions. So this is how the resistance is spread from one continent to the other continent over there. Now, the rise of the chloroquine resistance contributed to a worldwide increase of the malaria-related mortality, especially in the sub-Saharan African regions actually. Now, what is the mode of action of chloroquine? It has been found that the chloroquine basically it gets accumulated in the acidic digestive vacuole of the parasite. Thus, it starts interfering with the detoxification process of the heme. We know that the parasite develops on the RBC, which contains hemoglobin. Now, the hemoglobin is being divided into two forms, two major parts. One is the heme, the other one is the globin part. Now, the heme is the iron containing part which has to be detoxified in the form of a polymer. So many a times it is being released in the form of the hemozoin over there. So it is a byproduct of the hemoglobin degradation. While the globin part, which is the protein part, is being utilized by the parasites for its growth and multiplication. Now the parasites has to release these particular heme from its circulation so as to survive in the red blood cell. So they have developed a mechanism to release these particular thing. Now what happens is the chloroquine start getting accumulated in the digestive vacuole, thereby interfering in the detoxification of these hemes over there. Now the resistance parasite do not accumulate the drug to an extent that the sensitive parasite does. So this is the major reason which happens in the resistant parasite, due to which the accumulation cannot occurs in these resistant parasite, and the normal process of the hemoglobin degradation start taking place. Now it has been found that the gene which is very important to the chloroquine resistance is called as the PFCRT gene. Now the PFCRT gene is the gene which codes for a chloroquine resistance transporter over there, and it comes as a member of a superfamily of the drug metabolite transporter that lack the nucleotide binding domains over there. Now this is a image which is showing that how exactly the chloroquine is functioning. So we know that the chloroquine basically interferes with the hemoglobin degradation process, and in this hemoglobin degradation process the heme polymerization cannot take place properly because of the accumulation of these chloroquine in the vacuole of the parasites. Now there are various protein channels which are present, or the transporters which are present. So it has been found that there is a single point mutation which happens, due to which there is a replacement of a amino acid lysine by threonine at the codon position of 76 in the first transmembrane segment of these channels which are present in the food vacuole. So due to which the chloroquine resistance start taking place. So PFCRT has been designated as one of the marker to check the mutation for the chloroquine resistance. Now this is a diagram which is showing the chloroquine mutations over there and the PFCRT structure which is being shown over here. Now if we see, these are the major haplotypes which are being spread based on the codon position 72 to 76 across the globe. So we can see various haplotypes which are present in the India, Asian region, African regions and the South American regions. Now the next important history of the drug resistance came into the picture was the drug resistance against the sulfadoxine pyrimethylene drug. So when the utility of the chloroquine decreased across the globe, the scientists got interested why it is being decreased. So they found that there is an increased level of chloroquine resistance which has occurred. Then subsequently the sulfadoxine pyrimethamine got momentum to be used as one of the prime drugs for the antimalarial treatment, but over a period of time, due to high level of resistance to pyrimethamine spread rapidly across the globe, they have to be discontinued from many of the malaria endemic regions over there. So the sulfadoxine pyrimethamine resistance, if we see, has got a very interesting resemblance with the resistance which developed in the chloroquine, because both the regions overlap at that particular region. So it also started in the South Asian regions over there and then spread into the Amazon basin over there. Now this is a map which is showing the spread of the sulfadoxine pyrimethamine resistance across the various parts of the country. Now over the years, the WHO has accounted for the call for a resistance fighter, and in this call for the resistance fighter, they have come up with a novel strategy of artemisinin combination therapy. So WHO in the year 1998 came up with a WHO recommendation on the malarial treatment, which said that in response to the increasing level of the resistance to conventional antimalarial, the WHO recommends the use of the combination therapy. So in the combination therapy, what happens is one of the prime drug is being used, which is, for example, the artemisinin drug. The other drug can be affecting some other pathways or some other enzymes in the parasite. So what happens is, because of these things, there is a synergistic combination which start developing between these two drugs, and whenever these particular combination drugs are given to the patient, they act in a much more potent way than the single drugs over there or the monotherapy over there. Now what is the rationale for the use of this combination therapy? Now the rationale for the use of this combination therapy is basically to combat the increasing multi-drug resistance to the chloroquine and the other major antimalarials, which includes the sulfadoxine pyrimethamine over there. Then to increase the efficacy, which is greater and more potent than which is achieved by the monotherapies over there. To retard the development of the resistance against these individual drugs, and to use the synergistic or the additive combination partners against the artemisinin. So there are various artemisinin based combination therapies which has come into the market and which are being directed based on the clinical comparison of that particular areas over there. And another important thing is that because of this combination therapy, the load on the artemisinin has decreased, because we know that artemisinin production is a very costly affair. So what happens is, because of the decreased quantity of the artemisinin, the load has decreased. So the use of the artemisinin based combinations rather than the artemisinin monotherapies have been strongly recommended by the WHO. So nowadays most of the therapies which are being released in the market are basically the artemisinin combination therapy, which are being used for the antimalarial chemotherapy. So the artemisinin combination therapy has become one of the most important dosage for these artemisinin chemotherapy, and provides a fascinating opening for the development of new arsenals in the antimalarial brigades over a period of time. So this is a image which is showing that how the different forms of the artemisinin are being used, which are there in the market, and how effective they are. So we can see most of the artemisinin combination therapies are very effective, they are very potent. So what happens is, the clearance rate of these parasites is very high in the case of the use of the artemisinin combination therapy, because they are affecting the vital pathways or the processes within these red blood cell infected, erythrocyte infected parasites over there. And these artemisinins, they affect one of the major pathway which is called as the calcium homeostasis. So the calcium homeostasis plays a very important role in maintaining the structure of these parasites and the development and the multiplication of the parasite. Because this homeostasis is ruptured by the use of the artemisinin, the parasite dies immediately. Apart from that, the artemisinin has been found to affect number of vital mechanisms of the parasite, which includes blocking the heme metabolism over there. So the heme polymerization cannot occur properly. The parasite cannot utilize the hemoglobin to separate out the heme and the globin over there, and eventually the parasite dies. So the clearance rate with the use of the artemisinin combination is very high. Now, subsequently, in the last few years, there have been some alarming reports in which the resistance to these artemisinin combination therapies have been reported, especially in the borders areas of the Southeast Asia. So these are very alarming, and the scientists across the globes have now started working on these, checking up these particular reports, because artemisinin is one of the most important drug which is available in the market, and if it gets compromised, there will be a very tough situation created for the treatment of these malaria infected patients. So the scientists across the globe, they are working on the development and checking up of these artemisinin drug resistance reports over there, and simultaneously they are developing the new combination therapies which can replace or which can supplement these artemisinin combination therapy, ACTs. Now in this part of the lecture, so I discussed about the various anti-malarial drugs which are there in the market. So these antimalarial drugs are basically divided into various classes, which includes the chloroquine over there, the quinine which is there, the mefloquine. Then comes the folate mechanism affecting drugs, which includes the pyrimethamine, sulfadoxine. Then the peroxide class of drugs, which includes the artemisinin, the various classes of artemisinin, which are dihydroxyartemisinin, artesunate, artemether, which are available in the market. And then I discussed about the combination therapy. So how these combination therapies has been advocated by the WHO based on the reports of the already used antimalarial drugs. So we have seen that over the years from the 1940s, 1950s, various commonly used antimalarial drugs have circumvent into the resistance category. So there has been resistance against chloroquine, pyrimethamine, sulfadoxine, artemisinin. Reports are also coming in, but the scientists are checking these particular reports. And at the end, I discussed about what are the major key points which are causing the development of drug resistance against these particular antimalarial drugs, and how the parasites are developing the drug resistance. So with this note, I come to the end of this lecture. Thank you very much.